The objectives of work package 2 is to identify complex mixtures of concern for endocrine disrupting or immunomodulatory chemicals, where we want to identify complex mixtures, meaning very diverse mixtures that may be out there, chemicals that have not been identified yet, and how those chemicals act together. So the challenges with identifying complex mixtures is that we have two aspects that are relevant. First is that we have unknown chemicals from the exposure side, so we see many peaks, maybe in non-targeted analysis in our samples, but we don't know what those chemicals actually are. While on the effect side, we have effect data, but most often for the same chemicals that are often measured. The challenge of the prioritization is to find chemicals that have not yet been identified as relevant chemicals, and we provide tools for this or try to approach this in ENDOMIX. The assays we use in work package 2 are high throughput assays that target signaling assays, meaning for inflammatory signals, but also activation of nuclear receptors, such as thyroid receptors or AHR or other relevant receptors for the immune modulation, and as well as directly work with immune cells that we look at the differentiation of those cells and how they change their signaling and release of cytokines. The contribution of work package 2 is to provide information about the different mixtures that we could see. So we have both predicted mixtures as well as designed mixtures that we will distribute within the project, which will be the basis for the experimental testing in all of the different experimental setups. The main goal of work package 2 is to show that actually mixtures are really important and that it's the diversity of the mixture. So our plan is to, by targeting those complex mixtures, we can provide information on how chemicals can interact with each other, that the mixture interaction is important, and give an idea for regulators that really this mixture aspect is relevant to human health.